everybody, my name is Isabel Lehman, and today I'm going to be presenting my genetic disorder presentation on CPT1A deficiency. So, what is carnitine palmitoyltransferase type 1 deficiency, also known as CPT1A? I'm going to play the pronunciation for you real quick.
Carnitine Palmitoyltransferase.
So, carnitine palmitoyltransferase is a condition that prevents the body from using certain fats for energy, particularly during long periods without food, also known as fasting. It is considered a disorder of fatty acid oxidation, which is the process in which the body breaks down animal and vegetable fats for energy.
Carnitine Palmitoyltransferase.
Sorry about that. Now we're going to talk about the description of the disorder. The CPT1A gene is responsible for producing the enzyme carnitine palmitoyltransferase 1A, which is found in the liver and is essential for fatty acid oxidation, as we just discussed. Before we move on, I want to mention the fact that there is a CPT2 deficiency, which is why the 1 and the A are very important, and the CPT1A gene is the gene that corresponds to this particular CPT deficiency. Individuals with CPT1 are unable to break down fatty acids correctly as a result of a harmful change/mutation in this CPT1A gene, which severely reduces or eliminates the activity of the carnitine palmitoyltransferase enzyme. During periods of fasting or long periods without food, the long-chain fatty acids are important for the energy for the liver and other tissues, which is why individuals with CPT1 tend to experience the most symptoms during these periods without food. Next, we're going to take a look at some major characteristics of the deficiency. It is a rare disorder with fewer than 50 confirmed cases, and although it is not confirmed why, it is thought to be more common in the Hutterite and Inuit populations. Individuals with this deficiency tend to experience hypoketotic hypoglycemia, which is a combination of low blood sugar and low levels of ketones. People with the deficiency may also suffer from hepatomegaly. I'm sorry for that pronunciation, but basically an enlarged liver, liver malfunction, elevated levels of carnitine in the blood, and they are also at an increased risk for nervous system damage, liver failure, seizures, comas, sudden death, and they may experience muscle ache, muscle breakdown, muscle weakness, fatigue, etc. Now we're going to talk about the impact of the mutations on the structure and or functioning of the protein, also known as the molecular cause of the deficiency. The molecular cause is mutations in the CPT1A gene, which is found on the outer surface of mitochondria. There have been more than 20 mutations in the CPT1A gene found to cause this deficiency, and most of the mutations change just a single protein building block within the carnitine palmitoyltransferase 1A gene. Mutations severely reduce or eliminate the activity of the enzyme, as we discussed. And without the enzyme, carnitine is not attached to the long-chain fatty acids. So, as a result, the fatty acids cannot enter the mitochondria and be converted into energy, which is why reduced energy production can lead to some of the features of CPT1 deficiency, such as the low blood sugar and the low levels of ketones. Fatty acids may also build up in the cells and cause damage to the liver, heart, and brain. And the abnormal buildup is what causes the other signs and symptoms of the disorder, like the muscle breakdown and the fatigue, etc. Next, we're going to talk about the mode of inheritance. It is inherited, CPT1 is inherited in an autosomal recessive pattern. So, as we can see here, we have two unaffected individuals, meaning they are carriers of the trait. I have included this Punnett square. It's color-coded to kind of match this diagram here, so we can take a look together. So the two unaffected people who each carry one copy of the altered gene are big C, little c. That's the little c is the one copy of the gene. They're carriers. They have a 25% chance of having a child affected by the disorder, which we can see here and right here. I hope you can see my cursor, but the turquoise, basically. The chance with each pregnancy of these two carriers having an unaffected child who is also a carrier is 50%, which we can see by the darker purple highlights on the Punnett square and the darker purple children in this diagram over here. And the chance that a child will not have the disorder and will not be a carrier is 25%. So that is this lighter purple color over here, also on the Punnett square. So I have included another Punnett square to just further discuss this mode of inheritance. If only one parent is a carrier, meaning their genotype is big C, little c, like before, and the other does not carry the variant at all, because it is autosomal, it would be two big C's, as we can see in the red. So none of their children would develop the condition, because as we can see here, the chances of the children receiving two little c's is just not going to happen. That being said, the chances of having an unaffected child that is a carrier is 50%, as we can see represented by this orange over here. So before we move on to talk about the next thing, I have included the pedigree that I created this semester. As you can see, let me try with the red here maybe. As you can see, we have two carriers that have 4 children. Of their children, only one becomes afflicted with the disorder. And then of the next generation, we have 3 individuals who are considered to have the disorder, even though their parents do not necessarily have the disorder, except for, obviously, in this case, one of the parents does. So, in terms of genetic testing for CPT1, it's not necessary to diagnose the child with the deficiency. However, it can be helpful for carrier testing or prenatal diagnosis. And babies and children who are not treated for the deficiency can have learning problems, delays in walking, and other motor skills, and liver, heart, or kidney problems like we discussed. So early detection is important to prevent these severe outcomes of the deficiency. In terms of the options for CPT1 genetic testing, as we discussed, they are optional. However, they are available through blood sampling and DNA testing. Blood tests check for a lot of different things like elevated liver enzymes, elevated ammonia, carnitine in the blood, etc. The DNA testing, however, looks for the specific mutations in the CPT1A gene that are known to cause this condition. In addition to this, there is a special skin test that can be done that measures the level of the Carnitine Palmitoyltransferase 1 enzyme, and that's also a very widely used exam test. So before we conclude today, I wanted to provide you with a little bit more information on genetic testing. Parents are able to choose whether they want to do testing during pregnancy or wait until the birth to have the baby tested. Nonetheless, between 24 to 48 hours after birth, a newborn will undergo a state-specific routine blood spot screening to determine if they have one of many serious conditions, and most states offer this CPT1A screening as a part of this routine. That being said, the cost of the test does vary by state. Some states cover part of the cost, but most charge between $30 to $150 per infant. Although most of the states in the US do offer this CPT1A screening, there are available private labs or expanded newborn screening through private labs that you can have done as well. And healthcare providers and or genetic counselors will be able to help individuals locate these facilities. So that is all I have for you today. I have included the references I used throughout the entire semester. I very much enjoyed learning about CPT-1A. I hope that you all learned something new as well. And that's all. Thanks.